Namaste students, हमेशा खुश रहो बिटिया. I am JV'n's Dr. Neeraj Dholia, Faculty of Agriculture and Veterinary Science, Jayoti Vidhyapeeth, Women's University, Jaipur. So students, today we are here to discuss the topic that is chiral interactions. So this topic comes under the subject fundamentals of biological chemistry. So here we start. So what is chirality? Chirality is derived from a Greek word that is chir, that stands for hand. So an object is said to be chiral if the object and its mirror image are not superimposable, just like our right and left hand. Okay, so you can consider our right and left hand that they are mirror images, and when you put them one on another, they are not superimposed. Means the thumbs are outside. Okay, so they are not making a single image. Okay, so they are known as the superimposable, non-superimposable. So now you must be wondering what mean by the non-superimposable. When the mirror image of the object is placed over the original object and they do not overlap, then the object is its image and are said to be non-superimposable, as I explained. So the chirality was discovered by Louis Pasteur in 1848, and he observed that the two isomeric crystals were non-superimposable mirror images of each other. So they had the same physical properties but different in their ability to rotate plane polarized light. So this property was termed as the optical activity. So in non-polarized light, the electric and magnetic fields randomly orient in all planes. When the non-polarized light is made to pass through a polarizing filter or oscillator, get oriented in only one particular direction, and this is called the plane polarized light. So as stated that chiral molecules are optically active, means which implies that when a beam of plane polarized light passes through the chiral molecule, it interacts with the molecule in such a way that the angle of the plane of oscillation rotates. So here is the example that here you are having a chiral sample, and this is the plane polarized light. When it passes through the chiral sample, then this sample makes the plane polarized light rotate at a certain angle. So this is also an identification of the chiral sample. Is their sample having the optically active compounds or not? So how to identify chiral carbon centers? The rule of thumb is chiral carbon centers are carbon atoms that are attached to four different substituents that are placed at the corner of a tetrahedron. So the chiral carbon atoms are also referred to as the stereogenic carbons or the asymmetrical carbon atoms. Okay, so meaning is that a chiral carbon have four different valencies. Means in its all valencies, no one is repeated. Means all the four valencies are filled with different atoms. So that is a chiral carbon. So the chirality is present in the biomolecules also, in DNAs, in proteins, in amino acids, and they mix the various isomers like D and L isomer, R and S isomers. Okay, so that is important in the biological chemistry also. So thank you. Hope you are satisfied with this detailed session, and if you have any query, please write in the comment box. I will resolve your query. This session is powered by Digital Version 2, Jyoti Vidhyapeeth, Women's University, Jaipur. Thank you.